Okay, very good morning, my dear friends. Welcome to today's session in the regard of CPGET 2023 free online coaching for all competitive exams in plant sciences, along with CPGET, JL, DL, and Gurukula DL, JL, and PGT also, my dear friends. So, in this regard, today's our eminent resource person, huge experience personality, great botanist, more than 18 years experience, Dr.A.Kavitha Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College, Autonomous, Hyderabad. Madam is going to explain today's class in Pteridophyta, especially the type study of the Marselia and the life cycle of Marselia. So, my dear friends, listen carefully. If you have any doubt regarding of this class, please clarify yourself. And Madam is going to clarify your doubts. And listen carefully, my dear friends. And Madam, please welcome and start your session, Madam.
Right, sir. Thank you so much. Good morning, participants. Sir, am I audible?
Yes, Madam, you are audible.
Yeah. Right. So today we are going to start with the Marselia type study in Pteridophyta, the advanced forms of the sir, host to disable participant screen sharing.
Madam, 1 second. Sharing you, Nana. Nana. Chinni.
Isuna.
Okay, Nana.
Yeah. Yes, sir. Now it.
Yeah, Madam. Yeah, it's visible.
Right. So this is regarding the Marselia plant. Now, so when we talk about the Marselia, it is an aquatic plant among pteridophytes. So it is a semi-aquatic also, or coming out or growing in the marshy soil. You can see here, dam soil. So it is common in the temperate regions, and it always grows in the freshwater ponds and ditches. Now, Marselia quadrifolia, it is one of the most famous species. Why we are calling it the species named as quadrifolia? Because of the leaflets. You can see here, these are the 4 leaflets of the Marselia. So hence the species is named as quadrifolia. And Marselia munilia, minuta, they are commonly found in the other regions of the world. Now, when we talk about the general characters or the vegetative structure of the Marselia plant, now, so the vegetative plant, unlike the other pteridophytic plants, even here the dominant phase of the plant is the sporophyte, which is differentiated into the roots, rhizome, and the leaves. So you can look into this here, the structure of the plant. Okay, so this is the prostrate part of the plant, which is called as the rhizome, and these are the roots, and these are the aerial leaves. Now, so when you talk about the rhizome, roots, when I talk about the roots, you can see here, these are the adventitious or the fibrous types of root system, which are developed from the base of the nodes of the rhizome, and they help in maintaining buoyancy of the plant body. This is a free-floating pteridophytic plant. Okay, so the leaves will be coming out of the water, and the leaves will be floating on the surface of the water. So to maintain the balancing of the plant body, the roots are buoyant. They show the what we call it as the property of buoyancy. They are the balancing roots, just like in the pistia or another plants like nymphia, they have the balancing roots. All the aquatic plants, they have the balancing roots. Okay, here also the roots are fibrous and balancing roots. The next part of the plant body, that is the rhizome. So the stem is in the form of a rhizome here in the Marselia, and it has unlimited growth. So it will be growing along the surface of the water. So therefore it covers a very large area, occupying most of the water. And this rhizome, it is dichotomously branched, and it has nodes and internodes. And at each node, it will going to develop the adventitious roots on the ventral side. And on the dorsal side, from each node, it will give rise to single leaf. So you can see here, this is a nodes and internodes. This is the rhizome, which is dichotomously branched. These are the nodes. And hello, excuse me, please. You can mute yourself. Now, from each node on the ventral side, that is on the lower side of this rhizome, we can see the development of these adventitious balancing roots. And on the upper side, that is on the on the dorsal side of this rhizome, at each node, you can see that is just opposite to the roots. On the dorsal side, we can see the development of the leaves. Next, when we talk about the leaves here, the leaves of this Marselia, they are of compound. Okay, developing, that is the lamina is divided into leaflets. And each leaf, it is going to have a long petiole and 4 lobes, or we call them as carrots. Okay, so these are this is the entire leaf. This is a long petiole, and this lamina of the leaf is dissected into these 4 lobes. Now, so they are arranged in cross-like manner at the tip of the petiole, and each leaflet is triangular. You can see the shape of the each leaflet. This is each leaflet. It is having the triangular in shape, and the wings which are developing in this, they are in the reticulate manner. And the stomata, they are located on the dorsal side of the each leaflet and on the ventral side of these leaflets. So this is about the external morphology of the plant body, that is the Marselia. Now on this petiole, that is on the basal side of the leaf, at the time of reproduction, you can see the development of these specialized fruiting body-like structures. We call them as sporocarp. Okay, only this Marselia develop this sporocarp, within which the sporangia are going to be developed. This is one of the different type of pteridophyte we came across in the pteridophytes, because in Lycopodium and Equisetum, we have seen the sporangia, they are developed on the fertile leaves, which are called as sporophylls. And all these sporophylls, they are aggregated and forming the cone or the strobulus. But whereas this in Marselia, it is an aquatic plant, and the spores or the sporangia, they are developed in this fruiting body-like structures called as sporocarp. Now, what is the structure in the coming slides? I will tell you. Now, next we are moving on to the anatomy part of the Marselia. That is the internal anatomy of the rhizome. Now, the rhizome, the anatomy of the rhizome, it is going to consist of the 3 regions: the outer epidermal region, central cortex, and the internal stela region. Now, epidermis, it forms the outer covering. So you look into this here. This is the outer covering of the epidermis, single 1 or 2 layered. Now, below the epidermal layer, you can see this is the outer cortex. So the cortex is very wide here, which is divided into outer and the inner cortex, whereas the outer part of this cortex region, it is parenchymatous, and this parenchymatous tissue consists of large air chambers, which is often referred to as parenchyma. So which is the characteristic feature of all the aquatic plants. All the hydrophytic plants in the cortical region shows the presence of the parenchymatous tissue, and it stores the air. So which helps in plant body to float on the surface of the water. Now, the inner portion of the cortex, it is composed of sclerenchymatous cells. So this part, this is the inner cortex, which is of the sclerenchymatous, very thick walled, and this is the outer cortex region. And endodermis is present towards the inner side of the cortex. Next, the stele here in Marselia, it is referred to as amphiploic solenostele. That means to say it has a central pith in the region, and the protoxylem here is exarch in condition. So what is this amphiploic solenostele? So you can look into this here. The xylem, okay, it is divided into two ring-like structures. Okay, so this is the outer xylem and this is the inner xylem, you can see here. Okay, and also two pericycles, that is two steles we can look into here. So this is the outer endodermis and below that is the outer pericycle and this is the outer phloem, you can see here. Next comes to the, in between this outer phloem and the inner phloem, you can see here a ring of the xylem tissue. And internally, when we go further, it is again divided into inner pericycle and the inner endodermis, and centrally it is a pith region. Okay, so this type of, so this is a pith in the center, this is the inner pericycle, inner endodermis, a single row of xylem vascular, and then the phloem and the outer cortex, that is the outer endodermis and the outer pericycle. That is, what means to say here is the stela region is divided or it is differentiated into two steles. That's why this type of stele is called as amphiploic solenostelic condition. I repeat, this is another important point. In Marselia, that too in the anatomy of the rhizome, the stele is designated or it is described as amphiploic solenostele. Now, next we are moving on to the Marselia petiole, which is a very, very important part in the syllabus. Marselia petiole. In the practicals also, we are going to take the transfer section of this. Now, Marselia petiole. Petiole, stalk of the leaf. Now, even the petiole here, it is also differentiated. It is circular, differentiated into outer epidermis, inner cortex, and the stela region. That is the middle cortical region and the stela region. Now, when you talk about the epidermis, outermost layer, and it is single cell in thickness, and cells are parenchymatous and slightly elongated. And when you come to the cortex, so this is a single layer of outer epidermis, which is of parenchymatous and somewhat elongated and thick walled. Now, below to the epidermal region, we come across cortex, which is again divided into three parts. That is the outer cortex, middle cortex, and the inner cortex. Here also in the outer cortex region, which is present just below the epidermis, I can call it as the hypodermis, and it is going to made up of parenchymatous tissue here, which is divided into 8 chambers. Okay, this is the outer cortical region. Next, we come to the middle cortex. This lies below the, right. Okay, I'll show you this one. So this is a single layer of hypodermal outer cortex. And this, the 8 chambers, what you can see here, that is the middle cortical region, which is also made up of hypodermis. It is consisting of ring of 8 chambers, what we call it as parenchyma. Now, the 8 chambers, they are going to be partitioned by, so this you can see here, these are the 8 chambers which are partitioned by a single layer of the parenchymatous cells, which are thin-walled, resulting in the 8 chambers. Now, below to this middle cortex, the inner cortex, it is a very solid tissue, several layers in thickness, and these which contain starch and tannin inside them, and In the species called as Marselia minuta, some few parenchymatous layers are also present just inner to the middle cortex. So this is of the inner region, which are going to store the tannins as well as the starch. Now next, when we go for the internal part, that is a third region, that is the stela region. The stela region is triangular in shape, which is of protostelic type. Fifth region is absent, and you can see here the xylem is in the form of V-shaped xylem, and the projecting ends of this V consists of the protoxylem. Okay, metaxylem will be in the center, and the projecting ends. So you can see here, these are the projecting ends, which consists of the protoxylem, and it is represented as the protostelic condition. And the xylem here, it is surrounded by all the sides with the help of the phloem, which helps in conducting the food material. Now this is about the anatomy of the Marselia petiole. Single layer epidermis, a single layer of hypodermal outer cortex into 8 chambers with the longitudinal cells of this parenchymatous tissue. This is the inner cortex, many layered, storing the food material starch as well as the tannin. This is the central stela region, having a single layer of endodermis and pericycle. Internally is the, how we are identifying petiole. The xylem is in the form of V-shaped, a simple protostelic condition and surrounded by the phloem. This is about the anatomy of the Marselia petiole. Now, how we are differentiating Marselia anatomical rhizome as well as the petiole? In the petiole, it is a simple protostelic condition, whereas the anatomy of the rhizome shows amphiploic solenostele condition. Now, this is about the transverse section of the Marselia leaf. That is a leaflet when we talk about. So, the leaflet of the Marselia leaf, it is dorsiventral. The dorsal side, it is going to have the lower upper epidermis. Lower side is the lower epidermis. In between is the mesophyll tissue, which is differentiated into palisade as well as the spongy parenchyma. And you can look into this here. The epidermis, as it is the aquatic hydrophytic plant, the epidermal layer of the leaflet is going to be surrounded by cuticle. So this is a very, very important point, and it is also consists of the sunken stomata. Next, below to this upper epidermal tissue, you can see the parenchymatous tissue, which is the parenchymatous cells. They are long and very closely and linearly arranged, very close to each other, which is consisting of the chloroplast, which this tissue is considered to be as a palisade layer. And below to that, below to that, they are the round, thin-walled parenchymatous cells, which are very loosely arranged, which represent the spongy parenchymatous tissue. Even that is going to consist of the chloroplast for preparing the food material. Next comes just below, above to the lower epidermal layer. As this leaf is aquatic, so even here the mesophyll tissue consists of the parenchymatous tissue with a parenchymatous linear partition cells. Now in the center, we can see a single vascular bundle with xylem and the phloem. Xylem in the center surrounded by the phloem tissue. This is about the external morphology and the internal morphology of the Marselia rhizome and leaflet as well as the petiole. Yes, tell us any doubts? Any point to be repeated again here in the anatomy of the Marselia? Right. Next, we are moving on to the reproduction point of view. Now, at the time of reproduction, I told you here, in the external morphology. At the time of reproduction, these Marselia plant, at the base of the leaflet, we can see the development of some reproductive structures, or we can calling them as the fruiting bodies, called as the sporocarp. Now, what is the sporocarp? This sporocarp are the fruiting bodies which are going to consist of the sporangia, from which the spores are going to be liberated. Now, so the Marselia plant, when we consider regarding the spores, sporangial development of the spores, Marselia plant is heterosporous. That means it is going to develop two different types of spores in the sporangia. One are the megaspore and another one are the microspores. Two different types of spores are developed. Hence, this plant is called as heterosporous plant. Now, the spores which are smaller in size, we call them as microspores, and they are developed in the sporangia called as microsporangia. And the larger spore are the megaspore. They are produced in the megasporangia. So these two types of sporangia, they are found within the same sori or within the same stalk in the fruiting body, hard fruiting body like structures called as sporocarp. So I'll show you the structure here. So this is the sporocarp structure, right? Now, so these sporocarps, they are stalked structures. They are attached at the base of the petiole with the help of a short stalk, what we call them as the peduncle. So it is a bean-shaped like structure, and they are very hard and having a stony wall called as a capsule. You can see here. They appear, the shape of it, it appears like a bean shape. Okay, these sporocarps, they are having a stalk. This is the stalk which is called as the peduncle, and these sporocarps, they are developed at the base of the petiole. Right. So the wall of this sporocarp, it is a very hard and stony wall. Now, so this sporocarp, it has two internal chambers, and each chamber has a row of sori. So this is the one. Okay. So when we talk about the internal structure of this sporocarp, so this each one is called as the sori. So the complete sporocarp, it is divided into two chambers. This is a one chamber and this is a, so this is a one chamber this side and this is the right chamber this side. Now each chamber is going to consist of, each chamber has a row of sori. Okay. Now this is one chamber. All these are the sori. A row of sori. Now, so the sori of two rows, they alternate with each other. Now, the wall of the each sori is formed by its own indusium, and each sorus contains a row of megasporangium and several microsporangia. So, when we talk about here the structure, so this is a single sori, we call it as a sori, and each sori, it is going to consist of, right, so this we can clearly see here the microscopic structure. This each one is called as the sori. So this is one row of sori and this is another row of sori. So both the soris of the two rows, they alternate with each other. Now within the sori, you can see presence of a single megasporangium and laterally arranged two microsporangia. I repeat, each sori is consisting of a single megasporangium and a two or many microsporangium. Now, So there is a large placenta which is produced at the inner side of the young sporocarp. So the placenta of two sides, they alternate with each other. Megasporangium and microsporangium are produced on the same placenta, and this placenta is covered by the endosium. That is, each sori, it is covered by its individual outer wall layer, what we call it as endosium. So the megasporangia will mature earlier than compared to the microsporangium, and each megasporangium consists of a single megaspore. This is another important point to be noted. So I repeat this once again. Sporocarp. Within the sporocarp, there are two chambers. Within each chamber, there is a row of sori. Each sori is surrounded by a single layer of endosium, and internally to this sori, they develop the megasporangium and the microsporangium. Megasporangium consists of a single megaspore, and the microsporangium consists of, or it will develop around 32 to 64 microspores within the sori. Okay, so this is the structure of the vertical section of the sporocarp. Now, so how this sporocarp is developed? So it is believed that the sporocarp is a single folded pinna. Pinna is the leaf-like structure. It's a single folded pinna-like structure, has single vascular bundle, and the placentas are they produced on the vertical side. So on these receptacles or the placenta, sporangial initials are developed, which develop into the sporangium further. Now, dehiscence of the sporocarp now. Now, when the megasporangium is matured, it consists of megaspore, and the microsporangium, they develop the microspores. Now, so the mature sporocarps often are 2 to 3 years. That is, for the maturity of the sporocarp, it takes around 2 to 3 years. Now, after 2 to 3 years, I told you that sporocarp is surrounded by a very hard stony layer, which we call it as capsule. Now, that stony wall, it will decay, and the sporocarp will that is going to absorb the water, and the sporocarp will swell, and the sporocarp will split into two equal halves, and this it will absorb the water, and they will remain viable for a very long period of time. That is, they will get open and release the spores from the sporangium. So this is the development now. The microspore which is developed within the sporangium, they are around 32 to 64. Microspores are developed within the microsporangium. Now these spores, they will germinate and results in the male gametophyte. Now, so this microspore development you can see here. This is a microspore development developing into a male gametophyte. So this is a single layer of the microspore. First, it undergoes one unequal division, forming one small prothalial cell and this large apical cell. The apical cell will divide further and transforms into antheridial cells. Now, these antheridial cells will undergo periclinal as well as the anticlinal divisions, forming the jacket layer of cells, and at the same time, it results in the formation of androgonial cells. Now, once the androcyte mother cells or the androgonial cells are ready, the prothalial cell will disappear, and from this androcytes mother cell, each one will transform into anthrozoid. So here you can see a smaller prothalial cell, which will gradually reduce. The larger one divides into two antheridial cells. This antheridial initial will form three layered of jacket cell and a single androgonial initial. This androgonial initial of each antheridium divides and produces 16 anthrozoid mother cells. Each anthrozoid mother cell will transform into anthrozoids, that male gamete, which are melicoiled and single flagella. So this is very important point to be noted. A single flagellum will be there here of the antheridia, that is anthrozoids, and the prothelial cell will degenerate, and these anthrozoids, they become free in the surrounding water. Now the female gametophyte, that is how the female gamete will develop. So this is a megasporangium, this is a single megaspore. Now the single megaspore will divide into two cells. The smaller cell will occupy the whole beak and the larger cell does not divide further. The smaller cell functions as the apical cell. This is the smaller cell will functions as the apical cell, and this is the large prothelial or the nutritive tissue. Now this smaller apical cell will divide and cut into form a vegetative tissue. So this apical cell then functions as an archegonial initial. In this archegonial initial, it divides further to produce a primary cover cell at the top and the central cell at the base. So this is a cover cell at the top and the central basal cell. Now, so this cover cell which is at the top of this one will divide further and form neck initials, and they divide to form the neck region, and the central cell which is present toward the center, it transforms into an upper primary canal cell and the lower ventral cell. Now we can see the structure. It appears just like a venter or the archegonium. So this is the neck cells. This is the neck canal cell, and this one becomes a central cell consisting of the developing female gamete, the egg. So the primary central cell, it divides to produce a large oosphere, or we call it as a female gamete called as the egg. Now the ventricanal cell and the neck canal cell, they will degenerate or disintegrate when the archegonium will get matured, and it forms an opening for the entry of the anthrozoid. So through this here, the anthrozoids will reach this central part of the archegonium, and it will fuse with the egg and undergo fertilization. Now once the fertilization is done, it results in the formation of the zygote. Now the zygote will develop into the next stage of the development, what we call it as the sporophyte, the adult sporophyte. Now, so the oosphere or the zygote, it divides to produce 4 cells, 2 sister cells that develop the stem and the cotyledon, and other 2 cells they develop into the foot and the root part. Now the vegetative cells of the gametophyte, they form the calyptra. It is of 2 to 3 cells in thickness, and the calyptra will form an envelope around the developing embryo. Now the surface cells of this calyptra, they produce long rhizoids. The cotyledon and the root grow faster, and the calyptra comes and conic out of it. So like this is a calyptra. So these are the 4 cells. 2 cells will form the cotyledon, and 2 cells will form the root and the foot of the gametophyte. Now the root enters the soil. This cotyledon and the root, they grow faster, and the root will enters into the soil for fixation. The cotyledon will develops, it expands and develops into the first simple leaf. Now the primary root will get replaced with the help of the rhizome, that is adventitious roots, which grows horizontally and transforms into the rhizome. Okay, so this is the cotyledon developing into a single leaf. This becomes the rhizome part, and this is a foot developing the roots. These are the roots. So this is how the embryo is going to be developed from the zygote and developing into a young sporophyte. Now alternation of generation. Sporophyte and gametophyte here, they alternate with each other. So the vegetative part, the plant is a diploid sporophyte, and it is of heterosporous type, developing megaspores and microspores. The spores will germinate and form the haploid gametophyte. The gametophyte is dioecious. That is both microspore, that is the male and the female, both are separate from one another. Microspores will give rise to male gametophyte, and the megaspore will give rise to the female gametophyte. So both the gametophytes, they develop the gametes, they'll fuse and form the sporophyte, diplosporophyte again. So this is about the life cycle here. This is a Marselia plant. This is a sporocar, microsporangium, producing the microspore mother cell and the microspore, megasporangium, developing the megaspore mother cell and a megaspore. Megaspore will transform into male gametophyte. Microspore forms into male gametophyte. Megaspore develops into the female gametophyte. Male gametophyte consists of, they'll develop those antherozoids, the male gamete, and this female gametophyte will develop the archegonium, within which the female gamete is enclosed. These antherozoids, they move into this and fertilizes and develops a zygote. The zygote undergo division, resulting in this embryonal structure. This embryo, upon germination, that is upon development and differentiation, transforms into an adult sporophytic Marselia plant again. So a sporophytic, a diploid sporophytic plant alternating with the haploid sporophytic generation. So hence this life cycle is called as diplohaplontic life cycle. Okay, so this is about, I'm stop sharing here. So this is about the aquatic pteridophytic type study, the Marselia. Yes, any questions to be asked here? Questions, please.
Good morning,
Yes.
madam.
Yeah, good morning, Navya.
Madam, jacket cells present only in a microsporangium, madam.
Which one?
Jacket cells.
I didn't get you, please.
Jacket cells, madam.
Jacket cells. Yeah. So I'm just sharing the screen here once again. You can look into that. What are the jacket cells? Jacket cells are the covering cells. Okay, protective, the outer covering cells. So these covering cells you can see here in the archegonium. Okay, so you can see here.
Antheridium, madam.
Wait a minute. Right. So jacket cells you can see here. Right. The microspore, there is no such antheridium here. We can't say it as an antheridium because the single microspore, it is undergoing development and developing the androcyte mother cells inside it. And these androcyte mother's tissue is surrounded by these jacket layer of cells. First layer, second layer, and third layer. Okay.
Okay,
This
madam.
is indirectly, we can say it as, there is no peculiar structure of antheridium here. Okay,
madam.
microspore itself is developing and into the antheridium-like structure covered by the jacket layers. These anthrozoid or these androgonial mother cells or these androcyte, whatever we call, they are developing into the male gametes, the antherozoids.
Okay, madam. Thank you.
Right. Yeah, still any more doubts here?
Madam, dichotomous branch means, sir?
A single branch dividing into two branches. That is a single stem. Suppose if the rhizome is there here, rhizome is dividing into two branches.
Okay, madam.
We call it as dichotomous branching.
Okay, madam. Thank you.
Yeah, still any more doubts? Right, I hope it is clear. So with this, we have completed with the pteridophytes, the lowermost plants of the plant kingdom, which are called as cryptogams. So that ends with our cryptogamic study. Okay, sir. Tirupati sir, I'm done with this type study.